Hello, my name is Amanda Hake, and this past summer I spent time characterizing the role of microRNA in the functioning cerebellum. And in order to do this, I studied a knockout mouse, and we knocked out the enzyme dicer and looked at the effects. And as we saw in the mutant mouse, there was a loss of development and structure in the morphology. And I also looked at different markers for different cells, such as CD11B, and we found increased cell death in the mutant mouse because of the knockout in the dicer mouse. And I would also just like to thank the Ferlics for the opportunity to perform this research over this past summer.
Hi, my name is Virginia Barak, and I spent the summer researching the differences between cliff swallows and house sparrows in a serological study of the host-vector virus interactions and the differences they have because of their different life histories. It was a really cool project for me to do because I got to do research both in the lab and in the field, which exposed me to two different different sides of research that I enjoyed a lot. This project is important in the long term to kind of look at the interactions between hosts and vectors in birds, which also would help us look at interactions in humans and other sorts of animals. I just want to thank the Ferlics for giving me this opportunity. It's helped me learn a lot and given me many more opportunities since then.
Hi, my name is Elizabeth Bowes, and my research is on the black fly genome, and I was looking at some genes in the black fly genome that were specifically not insect related and looking at how those got into the genome, hypothesizing through horizontal transfer. So I really appreciate the opportunity to do this research from Dr. and Mrs. Ferlic. Thank you.
Hi, my name is Angelica Wu. I'm currently a senior, and my project was the characterization and expression of Similium vittatum, or black fly, silk genes, and examination of silk genetics and evolution. So in my project, I basically studied the black fly, and I characterized the genes, and by characterizing the genes, we hope to reveal some insight into perhaps biomedical potential, and so that's why I'm really excited about this project. So I guess I'd like to thank the Ferlics for funding this project and having a wonderful scholarship. Thank you.
Hello, my name is Nathan Messbarger from Kearney, Nebraska, and this summer I spent my time researching a new method for drug release. We're utilizing a reverse thermal gel for subcutaneous injection and microcapsules to release drugs. Thank you so much for your support this summer.
Hi, my name is Shweta Goswami, and I'm a junior biochemistry major at Creighton University. My project this summer was on the discovery of a mammalian riboswitch, and I performed several experiments such as equilibrium dialysis and inline probing in order to further characterize a possible riboswitch in mammals. Thank you so much, Dr. Ferlic, for funding me this summer.
Hi, my name is Chris Carlson. I'm a senior biochemistry major, and my project this summer was a study of the effects of a mammalian riboswitch on the enzyme gene and the spermine biosynthetic pathway. We chose to study this pathway because of its potential as drug development for cancer drugs. Previous studies in the Dr. Suckup's labs have shown the potential for a riboswitch, and this needed to be examined further in a more natural context. To do this, we created a new gene, including the introns, and examined this using a dual luciferase assay and qRT-PCR. The qRT-PCR results were inconsistent and will be studied in the future. The dual luciferase assay did not show a significant increase of the protein product. However, it did show that the gene was translated more often. And I would like to thank the Ferlics for their help in this summer research project.
My name is Anthony Edholm. I'm a senior from Andover, Minnesota. I'm actually a biology major here, and I've had the opportunity to work with the medical school and the Department of Medical Microbiology and Immunology to assess the correlation between Lynch syndrome and B-cell chronic lymphocytic leukemia. Lynch syndrome is one of the most common types of hereditary cancer. actually is the most common type of hereditary colon cancer, with 3% of colon cancers being related to Lynch syndrome. What we've seen in these Lynch syndrome patients is a development of a slow-developing leukemia known as B-cell chronic lymphocytic leukemia. Basically this summer I did a lot of immunohistochemistry and staining on different cell tissues, and from there we were able to kind of understand the relationship between these two cancers. So once again, this has been a wonderful opportunity. I've had a great summer and learned a lot, and I want to thank the Ferlics very much for the opportunity.
Hello, my name is Britney Bell. I did the Ferlic Scholarship, Summer Research Scholarship on the effect of warm acclimation on the gut and gills in Antarctic fish. In this case, it happened to be Trematomus bernaci. I'd just like to thank you so much for giving us the funding to do this, Ferlic, so thank you very much.
My name is John Otto, and the research I did this summer was Raman spectroscopic measurement of gas absorbance in organic aerosols. And I guess what we really learned from it was that we can, in fact, measure this absorbance, and so we're looking now towards making that a more concrete measurement. And I just want to thank the donors for letting that be possible.
Hi, my name is Rance Fujiwara. This summer I worked on studying the hydroxyradical reaction kinetics with organic aerosols in the atmosphere. So the main part of this summer was developing a experimental setup to perform laser-induced fluorescence to be able to determine the concentration of the hydroxyl radical in order to actually begin study of the kinetics, and the setup is right here. And I just wanted to thank the Ferlics, Dr. Ferlic, for the money and the opportunity to work this summer in the lab and make all the progress that we have done. Thank you.
My name is Kelsey Yamada, and I'm from Waipahu, Hawaii. I'm a senior chemistry and biology major, and the title of my project is Cloning Expression and Functional Characterization of Human Non-Canonical Ribonuclease A proteins. And basically what we're doing in this project is characterizing 5 new ribonuclease A genes, of which we recently discovered, but are different from the original, or the first 8 original ribonuclease A proteins. And the differences come in their catalytic sites, which are well conserved within the first 8, but not in the last 5, so ribonuclease 9 through 13. And so at this point in our project, we're at the expression stage. We've expressed it in, or we're ready to induce expression in the yeasts that contain this gene. And from there, we're going to test it functionally by assaying its biochemical properties, its substrate specificities, and its ribonucleolytic activities. And yeah, and I'd also like to thank Ferlic for providing me with the opportunity to do this project over the summer. It's really nice to be able to take time out of my year instead of studying to do research. And so it was really nice, and again, thank you.
I'm Ross Duvall. I'm a senior physics and math major at Creighton. My research is about possible dark matter decays into a light scalar particle, and this will help explain discrepancies between experiments, because they would expect to see a large quantity of antiprotons and anti-electrons, but they really only see anti-electrons. And so this will be explained if that dark matter decays into a light scalar particle that then decays into those anti-electrons. And we're going to use searches for fifth forces to limit these, to place constraints on the light scalar. And so I'd like to thank Dr. Ferlic for all his support for my research. And thanks.
My name is Wayne Gergens. I studied benzylated histidine residues on amino acids, and I started this process of synthesizing them via an intermediate that I optimized using different experimental methods, and then further experimentation yielded which isomers they were, because that was kind of a point of interest. And then we did another experiment to actually yield the histidine and then we are trying to put them in a peptide chain so that they can be incorporated into a 10 peptide chain so that they can be part of the CGRP receptor for a vasodilatory. And this project was really fun. It was good to work on. I'm very thankful for the Ferlics for letting me do this summer. Yeah, that's pretty much it.